Yeah, so welcome to this introductory course in biostatistics and bioinformatics. So my name is David Fenje, and I'll be giving some of the lectures in this course. And also Stuart Brown over there, he will be giving some other parts of the class. And also there's a third lecturer, Judy Zhang, who you will meet in probably next week, I think. And also, so this class, it will be lectures on Tuesdays and Thursdays at 2 p.m., almost always in this room, but there are some exceptions. On Tuesdays after the lecture, Pamela Wu, who you probably most of you know, will give a tutorial in Python programming. And she'll also help you to get started on the homework. So if you think you will need some extra help, definitely come and talk to me, and we can organize a tutor for you. So today I'm going to quickly go through the whole course in the first half, and then I'm going to talk a little bit about different ways that one can explore data. So as I said, so lectures are Tuesdays and Thursdays. So and the tutorials are right following the lecture on Tuesdays, and there is an extra tutorial this Saturday at 3 p.m. also in this room. So the location is almost always here except twice, so October 23rd and December 2nd. And then it's in the Skirball third floor seminar room instead. And there is a website that you can look all the information. So here is where we will update if there is new information. So you can, for example, download the slides from today's lecture, and then for Pamela's tutorial there are a few documents that you will use that you can also download from there. Then there is also reading material that we provide here, and also so when there is, for example, since this is on Saturday, it's been marked in red, and also when the lecture is in Skirball, we've highlighted that in red on the website. So always check there. That's where if there are any changes. So this lecture, so now I'm going to just go through the whole course quickly. And so you get an overview of everything that we're going to talk about. So this first lecture is about exploring data. So it will be looking at small data sets, how to best visualize them, and then going to very large data sets. So this example is 7 different samples that were analyzed with proteomics, and then it just shows how the different samples correlate with each other. So the second lecture, so the first lecture will only be displaying data in different ways. So it's only in the second lecture where we start to do some calculations on the data. We will calculate point estimates like mean and variance, and we will then also use these to go back and visualize the data in different ways. So for example, this figure shows how one can use a box plot to visualize a lot of information in a very compact way. So here what we have is we have 20 different measurements, these circles up here. In the box plot, what we show as a line here is the median of the data. And the reason to show the median here is that if we have skewed data, which it's not unusual that we have, it will be a much more robust measure. And then as a measure of the variance, we show the interquartile range. So we show a box that encompasses half the data points. And then we show the error bars as 1.5 times the interquartile range. And then one can also put a notch here on the box to show what the 95% confidence interval is. But we'll go into much more detail on the next lecture about these things. The third lecture, which Stuart is going to give, will be about different data types and representations in molecular biology. And there are a lot of them. There are many, many different ways to represent data. There are a few representations that are more common than others. So you'll learn about the FASTA file, which is a sequence, represents sequences. So you have a header line for each sequence, which has an identifier and then a description, followed by several lines with the sequence. And then you can have repeated blocks like this throughout. So this just gives you information about the sequence. Then the FASTQ file is, the Q stands for quality. So that has, in addition to the sequence, it also has a compact way to show the quality of the data. So each base in this case has a quality measure associated with it. And then there are other formats to describe, for example, protein mappings to the genome and other features that are mapped to the genome. And GFF3 is one other format. But as you'll see, there are many, many more formats, and there's always a lot of work that is involved in converting between different types of formats. So lecture 4 will be given by Judy Zhang. So she is in the Division of Biostatistics in the Department of Population Health, and she will lead you through statistics, and starting with probability. So as you probably all know, I mean, probability is a number between 0 and 1, that 1 usually represents that something that's certain, and 0 something that will never happen. There are also different ways you will learn about how, if you have two events, how to combine the probabilities of these events. If they're independent events, you can simply multiply the two probabilities. If they're not, then it's more complicated than that. You learn about conditional probabilities. What's the probability of an event happening if another event has happened in the past? And you'll also learn about Bayes' rule, which is very useful in a lot of different contexts later. So then there will be 2 lectures on sequence alignment. So this is a very commonly done in bioinformatics, is that you have 2 sequences of, for example, 2 nucleotide sequences, 2 protein sequences, and you want to find the best alignment between them, allowing for both insertions and deletions. There are several very good methods to do this well, and this is something that, again, Stuart is going to lead you through in more detail. So then lecture 7 will going back to statistics and talk about different kinds of distributions. So the most common distribution that we'll discuss is the normal distribution, and you learn about its properties and what the mean and standard deviation and so means for that distribution. But if you don't have a normal distribution, you can still if you have take a sample, you can still calculate mean and variance, but it will mean something very different depending on what your distribution is. Then an extension of sequence alignment is to search a database with a sequence. So here we have one sequence that we then compare sequentially to a lot of different sequences, a whole database of sequences. And here you probably heard about one of the most popular ways to do this is to use BLAST. And you probably have come across that already. But Stuart is going to tell you more in detail about how you should interpret the results and what the expectation value means in this case, and how and how to look at the sequence alignments. So experimental design is very important, and it's very easy to get it very wrong, and it's very easy to bring up examples where it really goes wrong, and I have come across many cases with people that have done some very bad experiments. So I'll try to go through about how to do good experimental design, and also give some examples of when it really went wrong. And it's always surprising. It's easy to laugh about it afterwards, but still, good scientists over and over do bad experimental designs. It's not at all unusual. So it's definitely worth spending a lot of time thinking about this. Proteomics Informatics. So this is going to be about mass spectrometry-based proteomics. So what we do there is we take our samples and extract the proteins, digest the proteins, and finally separate, make peptides that are reasonable size for mass spectrometry, and then separate them and analyze mass spectrometry. So what we do in mass spectrometry, we measure mass to charge. So usually we plot the spectrum as mass to charge on the x-axis and the intensity of the peaks on the y-axis. And the mass to charge nowadays can be measured really accurately, and we use that to identify the proteins. And then we use the intensity to quantify them. And we'll go into detail how this is done. So then, both with microarrays, proteomics, with a lot of different types of experiments, you can get information either about gene expression or what the protein concentration is. And people have worked a lot on how to analyze this, how to normalize it. So because from experiment to experiment you get variations, but you can apply different normalization methods and different analysis methods. And this is something that, again, Stuart is going to tell you more in detail about. Then we're back to more statistics. So estimation. So instead, one important thing is from a measurement, how do we estimate the different parameters? So we can either do a point estimate. So in this case, this shows just would be the average, would be the point estimate. But also other point estimates are the variance or the skewness of the curve. And then in estimation, we both estimate let's say the mean, but we also want to estimate the interval. So we want to get a confidence interval. And this is, there will be 2 lectures on this by Judy Zhang to how this is best done. Then again, more on sequences. So we earlier you had lectures about aligning two sequences and also searching databases with a sequence. But here now there will be how to align multiple sequences. And this is has its additional complexity when you have several proteins. So in this case, it's a casein from several different animals. And there's one region that we see here where one part here, we have a very good alignment for the different animals, but other parts there is less alignment, but there's still some residues that are very well conserved. And then the other thing, so that in statistics that you'll have a lot of use for is hypothesis testing. So this is just an example of someone who makes a hypothesis, says that the median age of the population is 50, and then you take a sample, and it turns out that the average age is 25. So then you reject the hypothesis. So this is the basis for hypothesis testing, but there will be 2 full lectures on this, giving you a lot of details and a lot of practice on how to do this best. Again, so we're back to sequence analysis, and now looking at sequence motifs. So here we've taken out a reasonably well-conserved region, and we want to look at exactly which bases are conserved and which are not. And there is one way that often this is done, which is very illustrative, is showing a sequence logo of this region. And it shows the information. So for example, when the letters are large, it's really dominated by one base in those positions. But here there's quite a bit of ambiguity. And then there are other places where it's dominated by one base, but it's really not. But quite often there are other possibilities also. So then another thing that you can use sequences for is to create different phylogenies. So this is of course based on that evolutionary. There was a common ancestor that would then in different species the sequences diverged, and so you can, by looking at how similar sequences are over different species, you can draw these phylogenies. Then back to statistics, so ANOVA, which is an sort of more generalization of the t-test, with applying to many different when you have many different groups. Then categorical data methods. So when you have different categories and you want to see whether there is any correlation between these categories, then you will learn about how to apply those methods. And then regression and correlation. So that's a very common thing that you will come across when you do experiments, is that you want to correlate different measurements and see whether there is any correlation between them. And then finally, non-parametric methods. So usually what we'll, throughout most of the statistics discussions, we will apply, assume that our population is normally distributed. But this is not always the case, and so you will learn about how to deal with that. The case when you don't know the distribution and you can't assume that it's a certain distribution. So then 24th lecture will be on signal processing. So it will be about the case when you can represent your measurements as as noise, and then you have a few peaks. So in this case, we can probably see that there are 3 peaks, but this last one is a little bit difficult to discern because it's very close to the noise. So what you learn is a general strategy how you can, from a measurement like this, how you can characterize and find and separate the signal from the noise. So so first, what you need to do is you need to know the properties and the distributions of both your signal and the noise, and then you can make a model of this, and then pick one detection method, and we'll go through a few different ways that you can do the detection. And then you can do, since you have modeled the data and model both the signal and noise, you can then simulate to test how to set the thresholds and the parameters. Then we'll learn a little bit about bioimage analysis, and that is how to take a microscopy image and extract information from it, and not just by looking at it, but automating this analysis and to be able to look at get much better statistics and look at many more images. So then next generation sequencing will be covered in one lecture, and this is probably most of you during your PhD will probably will probably be generating next generation sequencing data in your project. So this is will give you an overview of what kind of things one can do with that, how one can use it for confirming genes or finding new genes or finding variants. And so that's also another lecture that Stuart will give. Then there will be a lecture on machine learning, and basically what machine learning is that you make many measurements on, let's say you make a proteomics measurement where you measure thousands of different proteins, and you want to separate 2 cases, let's say disease from healthy. And so you want to, in this multidimensional space, you want to be able to find a separation between your 2 cases. So in this case, in a 2-dimensional case, we would draw a line to try to separate the blue squares from the red circles. And then finally, the last lecture will be on modeling and simulation. So often it is very good when you have a complex experimental data to try to think of a model that captures the essence of what you think is going on, and then use computer simulations to generate under the same conditions as your experiment, and then compare these simulations with the experiments to see if you can better understand your experimental data in this way. And this is can be very powerful, and we're going to go through a few really simple models. So the figure here just shows a time-resolved chip-chip experiment for replication fork progression in yeast. So you see, so it's time going down here, starting. This is before S phase. There's nothing much going on. When it enters S phase, you have some binding to the origins of replication. And then you have movement of the forks out, and finally it's over. So this just shows in 5 different regions what's happening, both in the experiment and in the very simple simulation model. So this was just the overview of the course, and then now I'm going to talk about a little bit exploration of data. Any questions on this part? Okay. So exploring data. So we'll start out. There is a data visualization blog that Nature Methods has put together. That's definitely worth looking at. It starts out with some very basic things like how to choose colors and how to general layout of presentations, and goes into some quite detailed detailed visualization methods. So I definitely recommend reading those. These are usually very short articles, but there's quite a lot of them. But it's definitely worth, because data visualization it comes naturally to some people, but for most of us it doesn't. But one can definitely work on it to improve it, because it's definitely important to do it, try to do it well at least. So here I've just taken a simple example data sets with 20 measurements, and this is the order that they were measured, and I've just simply, as a first step, plotted each point in the order that they were measured. So we see we have a little bit of a scatter, so but it looks rather random, and it looks like it's centered somewhere around 0 here. So this is a very good way to initially approach a measurement, because if something systematic happens, this is a measurement of the same quantity, so it shouldn't be anything systematic that after half the measurements there is a big jump or something like that. But this is probably a very simple and way to see if something really went wrong during the time that the measurements were done. So one way to then summarize it, because from just looking at these points, it's a little bit difficult to see what the distribution is. What we can do is we can make a histogram. So now this shows how many of the points were, how far they deviated from 0. And so now we see that if we have, depending on what bin size we choose, here we have just 5 different bins that we put it in, and we just count how many are in this area. So then we can, of course, change the bin size. And unfortunately for histograms, it's quite critical how you choose the bin size. And that's why they're not always the best way to do it. Especially if you have many points, it doesn't matter. You can get very nice histogram. But with few points, it's really, and 20 points is really too few, as you see, to really do a good. I mean, here we get a reasonable histogram, but it's really a little bit the bins are too wide, so we don't get a good feeling for what the data looks like. So there is another way to do this, and that's called the kernel density estimate. So now what we do is at each point here, so we put each point down here, and then we put a small curve around it, like a small Gaussian distribution, for example. You can use other things also. And then you do that for each point, and you add them up. And in this way, you get, first of all, you get a smooth curve. So you don't have the problem of that you have these discrete jumps. So later on, you'll see when we compare two different measurements, it's much easier to see differences when you have a kernel density estimate. And you'll learn later in the Python tutorials how to do this very easily. There are built-in functions to do both the histogram and these kernel density estimates. So the data was actually picked randomly from a normal distribution. So the red curve shows what the original distribution was that it was picked from. And you see that we can, with the kernel density estimate, we can reconstitute the original distribution reasonably well. Not perfect, it's only 20 points, but still. But with the histogram, we get these discrete jumps. So if we have more data, now we have 200 data points. We see that now the histogram is starting to work better, and we can. But again, we have to very carefully choose what width to use when making the histogram. But the kernel density estimate again will still be reasonably good. So this is again comparing to the original distribution. So now, let's say we want to compare different measurements. So here we have 5 different measurements. On the left side here, we have done 20 measurements for each of these values, and here it's 200. And then if we now show all the points here, we can see reasonably well what's going on. When we have 20 points, we see that there's probably no difference between the 2 first ones. The third one is maybe a little bit higher. Then this is the highest, and this is somewhere in between. And then that becomes very clear when we go to have 200 points. But then if we now instead compare either histograms or kernel density estimates. So this is the histogram that corresponds to this first 20 data sets, and then we're going to add on the other one. So this is, and that's the kernel density estimate for this data set. And again, this histogram here corresponds to that, and then the corresponding kernel density estimate. So now when we go to add on the second data set, we see that both from the histogram, it's rather difficult to see in this case what's happening, but it looks pretty similar. But when we do the kernel estimate, we see much clearer what's exactly happening. And now, remember, this lecture, we're only displaying data. We are not at all doing any calculations, so we're not going to be able to tell whether 2 measurements are different. That only comes next time. So then if we add on the third one, we see that from this histogram we really can't tell what's happening, but now we see a little shift in here, and then when we have many data points we also see the shift in the for the histogram, but clearer for the kernel density estimate. And then now when we go to this data set here, we see that there's a big jump. So it's almost completely separated, this distribution from the previous 3. And then again, when the last one will fall in between here. So again, it's a what I recommend when comparing different data sets is that if one has few data points, it's good to just show all the data points. That's probably the best thing to do. And also, if you have, in some cases, very few data points and other measurements have very many, it's good to show then also, and so that the reader can easily see that there is a big discrepancy in the number of measurements. So there's one more way. So we talked about histograms and kernel density estimates. But the third way that we can compare measurements is the cumulative distributions. So here now, what we do is we start at much lower point than any data point. And here we set that to 0. And then as soon as we reach one data point, we take a step, and then so on. So and then we end up, as we pass through all the data points, we end up to 1. And this is also very much clearer way to show differences when you have very few data points. So again, if we now, so this curve is for this data set here, so we can then go through to the next one, number 2, which almost completely is identical to that. And then the third one, which is a slight shift. The fourth one has a big shift, and then the last one is in between. So these are 3 different ways that we can quite easily compare data. Now, what I mentioned before is that sometimes strange things happen during measurements. And these are a few examples of that. When we only do a few measurements, it's a little bit difficult to see. But what we see in several cases, this seems to be, so now, yeah, I should probably mention that what I've done here is still, like in the beginning, plotted the data points in the order they were required. To see if there is anything. So here, what we see is that there seems to be a correlation between the order that they were acquired and but we can't see very clearly what the correlation is. Here one can see much clearer that these cases, it's probably a linear drift in the two, in 4 and 5. Here there's a big jump in the middle of the measurement, and here there seems to be a jump towards the end. But this one looks good. It's a randomly distributed over the whole, over the time that we measured. So in this case, it's much easier to see these kinds of systematic problems from showing all the data points as a function of how they were acquired. Because if we look at either the kernel density estimates or the cumulative distribution functions here, we won't see it as clearly. For example, here we see a little shoulder, but it's not. It's much clearer here what's happening. That it's at the end of the measurement something there was a shift. Again, here in this case we see that there's a bimodal distribution because we have really 2 different measurements. There was a big jump. But then when we look at these linear drifts, there is really no way to tell from the no easy way to tell from the kernel, either the kernel density estimate or the cumulative distribution function, that something has happened to the measurement. So definitely remember to, even though these plots of showing all the data points in the order that they were obtained are usually not so exciting, and that's the point. They shouldn't be exciting. They should be really boring and look random. So then, how to show different data? So the linear data is easy. So here we have a linear relationship, and then at low values we don't have any. We have some kind of threshold, so we can't measure below a certain level. So now we have done a few measurements on each here and shown them as black filled circles. But that's not ideal because they overlap and it's difficult to tell how many points there are. So what we can do is it's much better to use circles that are not filled. Here now we can usually see how many data points we have, much better than in this case. But even better, especially if you have the x-axis is categorical, so it's different categories. What we can add is a little bit of jitter and maybe show them in different colors. And now we really can see all the data points. So if we have an exponential function, that we should not show on a linear scale. It would, in this case, it would look like this. We just see the highest data points, and everything else gets squished down here. So that's, but we can simply take the log of that to the log of the y-axis if we have such an exponential relationship. And then we again can see the data properly. And definitely you should think a lot about how to how you can transform your data so that all the data, you can see as much of the data as possible in one plot. And these are some simple examples, but you're probably gonna run into other cases with your own data where it's not that straightforward. So then if we have a power function, again, in a linear scale, it's not, you can sort of see what's going on, but it's not as informative as if you take show both axes on a log scale. Because then you can see again that you have this linear relationship, but then it falls off, and there is a threshold, which you couldn't tell here because it was too small. So then if we want to illustrate correlation between two variables, we would show them as one variable on one axis and the other variable on the other axis. So this just shows this drawn from the same distribution. This is 20 points, 200 points, and more points. And as you can see, if you show it like this, it becomes they overlay on each other, and you can't really see structure when you have really many points. So one simple way that you can avoid that is to make these slightly transparent, so that it would. So this is the same data set shown with different levels of transparency. So you've just told the program to make each of these a little bit transparent, and then you can see the structure much better than you could before. And alternatives to this would be then to so here we have the all the data points shown in the same color without transparency and with a lot of transparency, or you can show it as a 2D histogram, as with a color scale on how many points there are in these areas. Or you can also show it as a 3D graph. But this is really not recommended, and usually they can look very pretty, but usually they're quite useless. So here's another example when there is a little bit more structure to this data. So if we just add a little bit of transparency, we don't see anything different. But if we add more transparency, we see that there is a feature here that's separate from the other distribution. And then if we show this as this is the case, the first case we looked at, and here is this other feature that we can see pretty well on the 2D histogram. And actually, in this case, even the 3D plot, since if we select the right angle, we can see that extra feature there pretty well. So now this is a proteomics data set that just 2 samples that are compared. And what we show, so first we show it on a linear scale. So these are the, it's a spectrum count, which is a way to quantitate, it's a simple way to quantitate proteomics data. So the 2 samples, there seems to be some correlation, but showing it on a linear scale is not so good as showing it on a log scale. So now we've simply taken the log 2 of the spectral count, and then we can see much more features. So what we see is that there are, since this is spectral counting, we see this discrete nature of the process. So and then we see that there's quite a good correlation, and there are outliers. But if you, it's probably not so clear, but if you look here, there is actually a lot of measurements that are not, a lot of proteins that are not found in one of the samples, but only in the other one, and vice versa. But those, since we take, those will be 0 in one case, so they will not show up here. But we could add them back into an arbitrary place. And so here we've just added them back at 0. So we can, so these are measurements that are not present in one sample, but present in the other one. So this is a way to give you, it gives a little bit more information also about, and observe that here we're also using the transparency so because it's quite crowded. So then we can do it some other way. So here we've shown one spectrum count against the other, but what we can instead take the log 2 ratio on the x-axis, for example, and we can sum the spectrum counts or take the average on the y-axis. And then again put back the ones that are only present in one sample at an arbitrary position. And then, because this would be easier than to see which ones are really outliers here. Or we can, this can be shown as a histogram. That will also give us an idea. So here we probably can see that there's quite a lot of proteins that are only present in one of the samples here, and less that are unique to the other sample. But here we very clearly see, can quantitate how many are there. So this is 2 samples. Then we have another example with 7 samples and 3 replicates. So the one thing that we want to make sure, if we have measured so many data sets, so these are proteomics measurements, so they each take a few hours to measure. So it might take a few days to measure something like this, and we want to see that things don't change during these measurements dramatically. So what we can do is to just make, again, a kernel density estimate of the distribution, what the intensities of our measurements are. So here we have shown all the, so these are 21 3 times 7 curves, and we show that they're reasonably well. But if we normalize it, we can get a better correspondence. But then in another case, we measure these 3 replicates, so that's the same. But then a few months later, we added another replicate, which was a little bit off here. But then with normalization, we can get it back, and we can then compare how we, I mean, we can then compare the data sets better. So then this is the same 7 samples. So here now, after doing the normalization, we show the different samples. So there's 7 samples here, and we compare them to each other. So it's actually symmetrical around the diagonal, and we see how well some of them correlate really well, and here we've also added in the correlation coefficient. We still show the proteins that are only in one sample, and then we can see that sometimes we get a really good correlation, and other times we have much more of a spread difference between the samples. But this is a very nice way to get a lot of data into one place, and that one can clearly see what's going on. And then we can, of course, do the same transformation as we did before to show instead the ratio, and then we can look for outliers that are shown in red here. Another useful way to give very large overview of data sets is Venn diagrams. And this can show some very high level information of what's happening. But it's usually worth paying a little bit attention to how you do the Venn diagrams, so that it doesn't, if you generate them automatically, they become usually very boring and not so useful. But in this case, we can very clearly see that some feature, like for example, the variants that we detect in RNA are much more than what we can detect on the protein level, but more than half of the ones that are detected on the protein level we can't detect on the RNA. So we can give, by designing the Venn diagram the right way, we can point the readers to see the interesting parts very well. Then, of course, you can do some very quite complex visualization, like in this case showing unsupervised clustering based on gene expression data, and then it has been decorated with all sorts of other information below. And so this is an example of a very complicated visualization that by the end of this course, you'll be able to do things like this very easily. Another few examples of going from very detailed information to very large overviews. So in this case, we looked at correlations between individual samples. So this shows the correlation. This is proteomics colon data, from 95 samples, and showing the correlation between mRNA and a protein. And it's usually a positive correlation, but it's the mean of the distribution is only 0.23. And this is very common, that one sees quite limited correlation between protein concentration and gene expression in a lot of different sample types. But another way to look at this would be to now, in this case, we're looking at copy number variation. So here is the different chromosomes. So on the x-axis is the copy number where it is, and then it's looking at what genes it affects. So now what we've done is, for this 95 colon samples, calculated for each location what the correlation coefficient is between between copy number variation and gene expression. And here it's for the same figure for copy number versus protein concentration. So what we see here is that we have these cis effects. So if the copy number changes locally, it will affect gene expression, which is not so surprising. But what we see, there are some things that are called transhubs that, for example, if the copy number changes here, it affects genes over the whole, over all the chromosomes. And there are several of these. And the same on the protein level. So this is, here we have taken a lot of data and condensed it in a way that we can easily see very large effects. So going from very detailed, so this is just aligning sequences to a genome to illustrate that at this location there is a variant. So it's a T instead of the references down here, it's a G. And so this is very detailed information. And also it gives us a little bit feeling for how much the error rate is. So here there are some sort of lonely mismatches, but these are probably just errors in the sequencing. So then if we go a little bit higher level, so this is chip data from Trypanosoma brucei, showing what the distribution is of histone H4 through the genome and histone H4 that's acetylated at lysine 10. And then what we can see very clearly in this case is that histone H4 that's acetylated at lysine 10 is very much enriched at the starts of these polycistronic transcription units. So this is one level up. So then we can go even another level up, showing genomic rearrangements. So here we have, in a circular way, we show all the chromosomes, and these lines show if there is a crossover in that we've detected in our sequencing data. So these are now, this is now very high level illustration of what's going on. Then another thing that we become more and more interested in is interactive visualizations. And because we have this enormous complexity of the data, but we really want to be able to jump between different levels. Just the same way as in Google Maps, you want to be able to zoom in and out and to see things. Ideally, we want to do that for both genomics and proteomics, and very easily. And so nowadays, the web browsers have become very powerful. So you can actually do a lot of this in the web browser. So this just shows one example of where you can look at different clinical data. So this down here is proteomics, and then looking at different stages of colon cancer, different parts of the colon, and then the same for the gene expression data in this. And then we have the up here the protein sequences, and then the DNA. So what we traditionally, so the genome browsers show this in a genome-centric view. So but the problem is that we're interested usually in what happens in the exons. But usually exons are small and introns are big, so it doesn't really illustrate. So what one can do, so this application lets you switch back and forth and just go down and, for example, remove all the introns at just with one click. Or maybe even better, not remove them, but shrink them and sort of just transform the space so that it enhances the information in the exons, but still keeps some summary information in the introns. And then we can even scale it more so that showing the parts that we were interested in. And then in this case, since we're interested in proteomics data, actually what we can let, each of these is a peptide that has some evidence in mass spectrometry. So what we can do is we can click on the peptide to then see the mass spectrometric evidence for that peptide. And we can, and this also can be brought up into the browser very easily. So we can go back and analyze these enormous amounts of data and choose the level of the level of detail that we want to see. So this is just some more details, and I'm going to skip that. So that brings us to the end of this lecture. So what I've shown you a little bit of how one can, starting from very simple data sets, how one can start investigating them, both using scatter plots, histograms, or kernel density estimates. How one can then compare these measurements using different, also these methods. And then we also looked at a little bit at how to illustrate correlation best, and then a few examples of other more complex visualization. And next lecture, which will be on Thursday at 2 p.m., will look a little bit more at how one can calculate different estimates of measurements. So I will stop there and take any questions. So Pam will take over now, and she will answer everything.